Next up, we've got ONTOFORCE, Carl Latham, as well as James Langdon from e-therapeutics. Please give them a warm welcome to talk about unlocking therapeutic discoveries. Thank you.
Thank you, Bruce. I'm really excited to introduce this presentation because it's a practical real-world application of how knowledge graphs combined with machine learning and artificial intelligence are really impacting drug discovery. But before I introduce James and e-therapeutics, just to introduce myself, I'm Carl Latham. I'm head of partnerships and sales at ONTOFORCE. At ONTOFORCE, we help life sciences companies to accelerate research and development by unlocking the power of data with our data and knowledge platform, DISQOVER. And DISQOVER is founded on semantic technology and an ontology-based knowledge graph. Joining me today is James Langdon, head of discovery biology at e-therapeutics. E-therapeutics integrate computational power with biological information to discover novel targets and develop new medicines in the field of RNAi in multiple therapeutic areas, including cardiometabolic diseases. And with that, I'll introduce James.
Thank you, Carl. Right. Okay, so I'm going to talk about the data, which is the foundation of the computational approaches that we apply at ETX. So what we're trying to do is model the complexity of human biology in order to identify novel gene targets and then rapidly design siRNAs against those targets. Ultimately, the aim is to greatly accelerate the drug discovery process. So just one slide on our platform. As Carl said, we develop siRNA-based drugs. This means that we are focused on diseases that we can affect by perturbing proteins in hepatocytes. siRNAs as a modality are now mature. This slide says that there are 5 approved drugs. It's actually out of date. The 6th was approved on Monday this week. This graph on the right-hand side just shows our platform and how it compares to our competitors. But the main thing I want to talk about today is the computational platform. We have HepNet. So HepNet is a combination of unstructured data, structured data, basically a knowledge graph, and we use ONTOFORCE for building the knowledge graph, and our proprietary network analysis. And ultimately, then we can wrap this all up in a nice graphical interface, and we can review the output of that, and I'll speak about that more in the next few slides. But the major outputs are gene targets, patterns, biological insights, the drug design, automation, what I mentioned on the previous slide. So, as I told you, the work that we do, a large part of it is based around trying to understand biological complexity. Now, often when biologists think about how proteins affect a phenotype, we think about the protein in isolation. But that's not how cells work. That's not how proteins work. Proteins interact with other proteins. When we think about how proteins interact with each other, we often view them in terms of a pathway, but even this is overly simplistic. When proteins interact with each other, they interact with complex signaling networks. So ultimately, then, this is what we're trying to do: build these protein-protein interaction networks and identify the most important nodes within that network that is driving phenotypic changes. And this approach is more likely to identify novel therapeutic targets because it actually tries to model how cells really work. On the basis of that, in order to build these network models, we need huge amounts of data. So where's our data come from? Well, it's a combination of written work, papers, grants, and websites. We combine annotations of proteins. So for us, typically, we're interested in proteins that are expressed and secreted from hepatocytes. We have information on non-coding RNAs, annotations of drugs and pathways, pattern, raw omics data, both transcriptomics and proteomics, and also genetic. We have information on diseases and real-world data as well. So we are integrating many different sources of data, both internal and public. And this has a number of challenges, and I want to talk about 3 of those today. So one of the major issues, and which is one of the reasons why we used ONTOFORCE for building the knowledge graph, is that building the graph is quite easy. But what's hard is getting the data into a format that you can build a graph. And one of the major issues that we have is ontology. So if you think about a gene often has multiple names. We have multiple databases that describe genes. Often the name of a protein will be different from the name of a gene. This is the same with diseases and pathways too. So in order to utilize all these different sources of data, we had to create our own internal ontology so that the entities within the knowledge graph are named consistently. A second major aspect is this idea of raw, can I go back? Yeah, of raw data. So when you describe, when data is described in text, you add bias. So when a scientist describes the results of an omics-based experiment, they're talking about results that they were interested in. They're not generally talking about the entirety of the data set. So in order then to reduce some of that bias, we can actually import the raw data as well directly. And so then the final aspect that I want to talk about is the use of the graph itself. So another issue that I think we have in terms of analyzing and interpreting the data is to know whether the data is good or not. So we have a reproducibility problem in biology. So how do we go about determining what makes a observation good or not? We can't really use the impact factor of the journal. As is well known, the pharmaceutical industry have tried to replicate data in high impact factor journals, and this is not always possible. In fact, in a large majority of the cases, it isn't possible. We also can't use the number of citations because this is ultimately linked to the impact factor of the journal. If the journal has high impact factor, it is more likely to be cited. So what we do is we actually use the structure of the graph, and we perform machine learning analysis of interactions between entities in the graph. So that ultimately then takes into account the structure, asking whether there should be an edge between these two nodes and how likely, for example, it is that this edge is correct. So in order to do this, we have to create true positive and true negative data sets. This is also hard because obviously in biology, we don't have distinct positives or negatives either. We know a lot about certain genes and very little about others. So what we do is we use a combination of what we call gold standard data sets, things like open targets for interactions between genes and diseases, but we can also do things like removing edges from the knowledge graph based on time. So we can remove, let's say, all of the edges we have from 2022 and then make predictions on the graph and try to see if we could recreate those edges that have been removed. So how are we using this data? So ultimately what we can do is we can generate hypotheses using machine learning on the knowledge graph. We can then take those disease gene, for example, predictions, and we can build networks around that. What you then do is you create a target triage problem, because we can ultimately run this over millions of gene disease pairs. We can do it for every disease in the knowledge graph, hundreds of them, all diseases we can potentially affect by perturbing genes in hepatocytes. So what we're going to be building, what we are working on now, is generating large language models that are able to then triage this long list of potential gene disease pairs. And then we have automated sRNA design as well. And what this means is that we can greatly speed up the drug discovery process. So we can go from identifying a target to IND enabling or at the point where we would want to enter IND within 12 months. And we're actually doing that. So we have 4 preclinical pipelines, and you'll be hearing more about those next year as they come to fruition. And now I'm going to hand back to Carl for the final part of the presentation.
Thanks, James. Yes, so as James kindly shared, that the Discover knowledge graph is part of the HepNet ecosystem, and customers can benefit from 140 public data sources that we pre-ingest into the Discover platform. So sources like clinicaltrials.gov, Open Targets and Chembl, and also the public ontologies that James mentioned, things like ICD-10, ICD-9, MESH and SNOMED. Discover also has a fully customizable user interface, which can be adapted according to specific use cases and is used by a wide range of life sciences professionals within biopharmaceutical organizations. And today, the way in which we deliver Discover to our customers is that we have 4 solution areas across the entire drug development lifecycle and 9 pre-built applications. One of these out-of-the-box applications is target identification. And with the target identification application, you can access the pre-processed public data we have from sources like Mondo, Open Targets, as I mentioned before. And you can integrate your internal data, so things like ELNs, target repositories. And finally, we also have a number of pre-built connectors and pipelines for third-party sources like OffX, Clarivate, Cortellis, and also BioRelate. And I'd just like to finally share, we started working with a German pharmaceutical company about 12 months ago that deployed this target identification application. And one of the targets that they had was to identify four new targets a year. But they had an 80% rejection rate on targets at the scientific board level. And since they've deployed the DISQOVER for Target identification application, they've been able to reduce the amount of time spent on collating information on targets from months to a matter of days. And also using the link prediction within the knowledge graph, they've been able to identify targets that they hadn't been able to identify before. And if you're interested in learning more about this application or any of the other applications that ONTOFORCE have developed, we're at booth 809, and one of our team would be happy to provide a demonstration of the product to you.
Thank you both. Any questions for the team? Oh, we have one back there. You can use that mic right there.
I have just a quick question to you, James. Is your knowledge graph all based on NLP or also some kind of curation?
So I didn't quite hear that. You were asking about the knowledge graph?
Is it all based on NLP, what you're doing?
No. Okay, so natural language processing is used to extract information from text, but then it's also additional quantified data as well, and simple databases like the expression of genes in hepatocytes, for example. So it's a combination of things.
Any other questions? Oh. Oslo?
Thank you so much. So the knowledge graph itself, is it only specialized for the hepatocyte data, or do you have other cell information in there?
Okay, so we do. So obviously for us, we're trying to develop drugs, and these drugs are hepatocyte specific. But the knowledge graph contains information between the flow of signals between hepatocytes and other cell types and tissues. You can imagine there are going to be ligands secreted from hepatocytes that bind to receptors on distant cells. So we have that data too.
May I?
Please, we've got time
Okay.
for one more question.
Thank you. So I assume that you're using graph neural networks of some sorts, like convolutional ones. And not all data is as useful as other data sets, obviously. So did you recover or did you identify any data in the knowledge graph that turned out to be not that useful? And are you stratifying for that?
Okay, so ultimately that's what the graph convolutional neural network should identify, really. I think this is one of the points I had. It's very hard to know whether data is good or not prior to ingesting it into the graph. My solution to that, and we'll find out if that's a good solution or not in the future, is to add all data, as much data as we possibly can, so that then by using a graph, you've got weight of knowledge around the interaction between two entities that potentially then support that. And that's ultimately then what we're trying to do with the graph machine learning.
So thank you, James. Truly appreciate the insights.